Back, y'all. So let's do the flashcards for the chapter 2 in biology. Let's start. Eukaryotic cells reproduce via. So when it comes to reproduction, we are talking about. Are we talking about sexual reproduction or asexual? Because with asexual it's mitosis, with sexual it's meiosis. So we are talking here about mitosis, asexual reproduction. So we have DNA replication. So these chromatids, so these are haploid, they turn into a diploid, and then we have the mitosis happening, and the DNA replication is in the S phase, right? And then we have two diploid cells. So what does it mean? You have two diploid cells. These are diploids, and these are, so this is kind of what can confuse us. Mitosis, yeah, not gonna work for us. So let's ask a question. What is the ploidity? Is it ploidity of the chromosomes during the cell growth cycle and mitosis? So S stands for synthesis. That's why we have DNA replication. Before that we have the G1 phase. The cell is in the G1 gap 1 phase after cell division. Mitosis is complete. The DNA is in a diploid state, meaning it has two sets of chromosomes, 2N, with each chromosome consisting of two sister chromatids. I don't know why we are not seeing that here, so that can be weird. Then each chromosome is duplicated, resulting in two identical sister chromatids held together by a centromere. However, the ploidy remains diploid because the number of sets of chromosomes doesn't change. In G2, the cell prepares for mitosis. The DNA is still diploid, with each chromosome having two sister chromatids. So each of these chromosomes has two sister chromatids. Here it is one sister chromatid, one chromatid. Then we have the mitosis taking place. So we have the prophase, metaphase, and anaphase. So in prophase, the chromosomes condense, becoming visible under a microscope. Each chromosome consists of two sister chromatids, right? So we have the replication, each chromosome has two sister chromatids, right? The nuclear envelope begins to break down. The ploidy is still diploid. Chromosomes aligned at the cell's equator. The centromeres are attached to spindle fibers. So what are the centromeres? Let's take a look at the centromeres for a second. So this is the centromere, this is the telomere, and each of these are chromatid. So chromosomes, the centromeres are attached to spindle fibers. These spindle fibers are coming out from the centrioles, right? So the attachment of the spindle fibers happens at the metaphase. They meet in the middle, and the ploidy is still 2N, diploid. The sister chromatids are pulled apart towards opposite poles of the cell. The ploidy changes from diploid to haploid. So it is during the separation, during anaphase, that the cell goes from diploid, having two sister chromatids, to a haploid, as each separated chromatid is considered an individual chromosome. And then we have telophase, the separated chromatids, now individual chromosomes reach opposite poles, nuclear envelopes reform around each set of chromosome, the ploidy remains haploid. And then you have the cytokinesis. After mitosis, the cell undergoes cytokinesis where the cytoplasm is divided, resulting in two daughter cells. Each cell is now diploid because it has a complete set of chromosomes. So regardless of it being one chromatid or two chromatids, each daughter cell is now diploid because it has a complete set of chromosomes, right? See, the only place that it is haploid is in anaphase, right? And telophase as well. But then after so when the cytokinesis happens, it is back to diploid. So how about meiosis now? Meiosis is a specialized type of cell division that occurs in sexually reproducing organisms and leads to the formation of gametes, sperm and egg cells. Meiosis involves two consecutive divisions known as meiosis 1 and 2, resulting in production of 4 haploid cells, gametes. The overview is prophase 1. The chromosomes condense in homologous chromosomes with the same genes but potentially different alleles pair up in a process called synapsis. This stage is marked by crossing over exchange between, at the end of prophase 1, the nuclear envelope breaks down. With the key difference that the starting cells are haploid rather than diploid. Chromosomes condense in a new spindle apparatus forms. Does something special happen before meiosis 1 that does not happen in mitosis? Are these specific cells, so like gametes, right? Occurs in sexually reproducing organisms and leads to the formation of gametes, sperm and egg cells. These cells like a gametocytes that do meiosis. Where does this take place? In our sexual organs? Yes, several unique events occur before meiosis 1 that distinguish it from mitosis. One critical event is the pairing of homologous chromosomes in a process called synapsis during prophase 1. This process does not occur in mitosis. Here are some key features. Synapsis and crossing over. Homologous chromosomes pair up. Reduction in chromosome number. The separation of homologous chromosomes during anaphase 1 results in a reduction in chromosome number. The cells produced after meiosis are haploid, meaning they have half the number of chromosomes as the original cell. Independent assortment. During metaphase 1, homologous chromosome pairs align randomly at the cell's equator, leading to independent assortment. Is this the same as crossing over or recombination? This random orientation contributes to genetic diversity among the resulting gametes. So is independent assortment different than crossing over or recombination? Or is it the consequence of that? These unique events are crucial before meiosis 1 for generating genetic diversity among the gametes, which is essential for the variation seen in offspring. As for where meiosis takes place, it occurs in the germ cells, specifically in the cells that give rise to gametes. In males, meiosis takes place in the testes within specialized cells called spermatocytes. If you remember, spermatocytes, we have primary spermatocytes and secondary. So, and before that, they are spermatogonia, ultimately leading to the production of sperm. In females, meiosis takes place in the ovaries within specialized cells called oocytes, leading to the production of eggs, ova. Or ovum is a single egg. The process of meiosis is central to sexual reproduction as it ensures that gametes have half the chromosome number of somatic cells, facilitating the restoration of diploidy during fertilization. Independent assortment and crossing over are related processes that occur during different stages of meiosis. While they are distinct events, the consequence of independent assortment is an increased genetic diversity among The consequence of independent assortment is an increased genetic diversity among gametes, and crossing over is one mechanism contributing to this diversity. Independent assortment occurs during metaphase 1 of meiosis. It refers to the random orientation and separation of homologous chromosome pairs at the cell's equator. The way these pairs align is independent of how other pairs align. Independent assortment results in different combinations of maternal and paternal chromosomes in the gametes. This random assortment contributes significantly to the genetic diversity of offspring. We have crossing over, recombination. Crossing over occurs during prophase 1 of meiosis. It involves the physical exchange of genetic material between homologous chromosomes. This exchange takes place at the chiasmata, the points where chromatids of homologous chromosomes are crossed. Crossing over occurs during prophase 1. However, independent assortment happens in metaphase 1. Crossing over recombination involves physical exchange of genetic material between homologous chromosomes. But independent assortment is the random separation of homologous chromosome pairs. So these chromosomes here could be, we'll come across that. But we definitely need to put independent assortment here. Random separation of chromosomes. At equator. Okay, nice. Let's continue with the flashcards here. During what cell cycle phase the cell synthesizes mRNA and proteins? I believe this is, so when is it after mitosis or before mitosis that you need to synthesize mRNA and proteins? So once your replication is done, basically you have done your transcription in the central dogma picture, then you will do the mRNA and protein synthesis. So you have the S phase And after that, you basically should have the G2. So G1 is after the mitosis. So you need to do it again. It's a cycle, right? And this is to prepare for subsequent steps leading to mitosis. So you do G1, S, G2, and M. And G1 stands for gap 1. So there we have it. We have also a G0. That's when you don't want to replicate. So a cell will enter what cycle phase if it does not need to divide? That's G0. At the what checkpoint cells choose whether or not to divide? That's the G1 checkpoint, right? So we have the G1 checkpoint restriction. We have the G2 checkpoint. P53 is in charge. Let's actually add this here. What is in charge of G1? Oh, what is in charge at this checkpoint? DNA is replicated in what cell cycle phase? That's S phase. S synthesis. In what cell cycle phase cells grow and make organelles? We have the G2 phase. Organelles, G2. At what checkpoint the cell will check that the DNA has replicated correctly? It should be the G2 checkpoint. Replication being done correctly because G2 is after S, so it makes sense. Mitosis and cytokinesis occur in what cell cycle? As the interphase, M phase. So both cytokinesis and mitosis happen in M phase. The 4 phases of mitosis are PMAT. Interphase precedes mitosis. It is not part of mitosis. So what is happening? Interphase is when the centrioles have not even started to separate, right? Interphase G2, repair of replication errors. Nice. Duplicated centrioles. Nuclear membrane is still intact, and duplicated chromosomes are there. But then you have the prophase where chromosome condensation happens and creation of mitotic spindle. You have the microtubules. You don't have that in interphase. You have condensed chromosome. You can see the chromosome. You cannot see the chromosome in interphase. And the mitotic spindle is there. What else? You have the kinetochore. But then you have pro-metaphase. So Mat's pro player, anaphase, anti-anas. Then disintegration of nuclear membrane in pro-metaphase. Kinetochore attaches to mitotic spindle. The kinetochore attaches to the mitotic spindle. And in metaphase, the chromosomes are maximally condensed, aligned on the equatorial plane. In interphase G1, we have synthesis of proteins and cell organelles. PMAT, let's go, it's like goes from here to here. So anaphase, separation of sister chromatids. Early telophase, sister chromatids grouped at cell poles. disintegration of the spindle apparatus. Late telophase, chromosome decondensation. Creation of, so the difference between early telophase and anaphase is that at early telophase, these chromatids are at the poles already, but in anaphase, they're just separating. And in late telophase, the chromosome is decondensed. You have the creation of nuclear membrane. You have cell division on the equatorial plane. And in interphase G1, so we have interphase G2, which is right before the PMAT, the mitosis. But interphase G1 is after, which is the synthesis of proteins and organelles. So you have the centriole, one centriole in each cell, daughter cell, and single chromosome. Beautiful. And we got this question right. Interphase includes what cell cycle? Interphase includes, honestly, I would go G2. G1S and G2. I want to say both G1 and G2. So interphase is all of them. So it's G1, S, and G2. Phase where the cell spends most of its life. This is most of the life for the cell. Again, in what mitosis phase the DNA condenses, centrioles migrate to opposite poles, and microtubules form. This should happen in the prophase because I say so. In what phase chromosomes meet in the middle? Of course, metaphase. At the metaphase plate. Mets, players lining up. In what phase sister chromatids separate? Anaphase for separation and move to the opposite poles. Anaphase apart. What do we have for Anna? Anti-Annas, twin sisters, tearing pretzel. Too hard for me, I don't like that. In what phase chromosomes decondense, the nuclear membrane forms and cytokinesis occurs? We know this is in telophase, and keep in mind that the nuclear membrane forms in telophase. And what do we have for the tearing tape, telephone, newly sealed box, bullshit. What number, the number of complete sets of chromosomes in a cell is referred to as the ploidy, like complete sets is 2n diploid, ploidy of a cell. The number of complete sets. So haploid is n, diploid it's 2 complete sets. So if you have 1 complete set, it is haploid. The phases of meiosis, PMAT1 and PMAT2, in between them we have interkinesis, right? Recombination occurs, homologs pair, sister chromatids remain attached. I know you can't probably see this because it's hard to see it, but DNA is copied, 2n, recombination occurs, mitotic spindle, chromosomes pulled apart, and phase 1, cell pinches in the middle, 2 daughter cells, chromosomes line up at the equator, sister chromatids pulled apart, cell pinches in the middle, 4 granddaughter cells. Haploid, diploid. At the end you get haploids, but for mitosis, at the end you got diploids, right? And if you look here, you have a dark blue and a light blue. You copy that, so you have a diploid dark blue and diploid light blue. So that was for interphase, and now it's prophase. Now you have the recombination happening. You see it kind of it gets crisscrossed, you know, like you have some sections of the light blue as dark blue, some other sections again. Then you have telophase 1 inside the kinesis, and now you come again for prophase 2, and these two cells do the mitosis again, and that's why you get these four. So at the end you have 4 haploid cells at the end of 2 mitosis cycles, as known as meiosis. And you have one recessive, light blue. You have one dark blue, completely dominant, and 2 these are homogeneous, and 2 heterozygous granddaughter cells, haploid granddaughter cells. What is when sister chromatids don't separate properly during anaphase? This is non-disjunction. Results in aneuploidy. So non-disjunction is 2n plus 1 and 2n minus 1. It was supposed to be 2n and 2n, right? So one is 3n, the other one is 1n. And you can see homologous chromosomes, non-disjunction, and we have n minus 1, n minus 1, and then we have n plus 1 and n plus 1. Let's add results in what? Aneuploidy. So the sister chromatids don't separate properly during anaphase. What is when 2 chromosomes of a homologous pair exchange segments with each other? This is recombination, crossing over. Synapsis, pairing of homologous chromosomes. When they pair, it's synapsis. And then when they're crossed over, Marvel Infinity War is the most ambitious crossover event in history. Homologous chromosomes aligned, chromosome crossover, recombinant chromatids, non-recombinant chromatids. So these two, like those 2 heterozygous ones, at the end of the day, those are the recombinant chromatids, and we have also non-recombinant chromatids. So you can see here the event and outcome. So something happens during each phase. This is a good way to look at it. So whatever happens, there's an outcome for it. So if the event for the S phase, the interphase, is chromosomes are duplicated during interphase, so that's the event, the resulting sister chromatids are held together at the centromere. The centrosomes are also duplicated. So the outcome of the duplication of chromosomes during interphase is that the resulting sister chromatids are held together at the centromere, and the centrosomes are also duplicated. Again, centrosomes with centriole pairs. Chiasmata is here. Okay, wait a second. So centromere is here. And what is a centrosome? What is the difference between centromere and centrosomes? We expect that the centrosomes be the 2 chromosomes, right? But centromere is a specialized region on a chromosome that plays a crucial role in cell division. It is responsible for the attachment of spindle fibers, facilitating the separation. Centromere acts as attachment site for mitotic or meiotic spindle fibers. It ensures that the accurate segregation of chromosomes, and it is involved in formation of kinetochore, a complex of proteins that binds to the centromere and interacts with spindle fibers. What was kinetochore again? Let's see. Here, homologous chromosomes are attached to spindle microtubules. So these spindle fibers are microtubules. They're made out of tubulin. If you remember, tubulin is like a dimer. So in microtubules, we have 13 alpha and beta unit tubulins, right? And so the homologous chromosomes are attached to spindle microtubules at the fused kinetochore shared by the sister chromatids. Now, what is centrosome? Centrosome is an organelle in animal cells that contains two centrioles. Ooh, nice. Centrioles are cylindrical structures composed of microtubules. The centrosome is often considered the main microtubule organizing center in animal cells. So centrosome, you should not, it is not related to like the whole chromosome structure. It is an organelle that has the centrioles inside it. This is very important. So they play a crucial role in organizing the microtubules that make up cells' cytoskeleton. During cell division, the centrosome duplicates and the two centrosomes move to opposite poles of the cell, forming the spindle apparatus. The spindle apparatus, in turn, helps separate chromosomes during mitosis and meiosis. Beautiful. So not only chromosomes are duplicated, centrosomes are also duplicated, which consists the centriole, and the resulting sister chromatids are held together at the centromere. We have two sister chromatids that are held together at the centromere. Now, this is the event, right? Now we have prophase 1, which is, you can see these are duplicated. You don't have the nuclear membrane anymore. So chromosomes condense. The nuclear envelope fragments. You don't have it anymore. Homologous chromosomes bind firmly together along their length. So they bind. What is this binding called of the homologous chromosomes? Crossing over. The synapsis is the pairing. So the pairing, the binding is synapsis, forming a tetrad. Chiasmata form between non-sister chromatids. So we have sister chromatid. So sister chromatids, if you can see here, is one blue and one red, right? Two blues are sister chromatids. But now, the non-sister chromatids have something called chiasmata. Let's see. This should be the chiasmata, right? Chiasmata, okay. There you go. These are chiasmata. It's the two non-sister chromatids. Crossing over occurs at the chiasmata. Okay, that's when you have the crossing over at the chiasmata. Spindle fibers emerge from the centrosomes. And from the centrosomes, the spindle fibers emerge. Then you have the homologous. This is the holy grail. Then you have the at pro-metaphase 1, we have the homologous chromosomes that are attached to spindle microtubules at the fused kinetochore shared by the sister chromatids. So we have the centromere with kinetochore. So these kinetochores are at the centromere. I believe there was something here about kinetochore. There you go. The complex of proteins that bind to the centromere and interact with spindle. But kinetochore mitotic checkpoints is a large proteinaceous structure that mediates interactions between chromosomal DNA and spindle microtubule polymers. More than 80 kinetochore proteins have been identified using various genetic, functional, cell biology, and proteomics approaches.
This visualization isolates a single chromosome at mitosis. The chromosome contains 2 identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
Man, the animation creator for this video, he needs a raise or like an Oscars or something. Drew Berry, man, props to you. Walter, Eliza Hall, yeah, you guys rock. And we can even animate the process. Is still dumbfounding and impossibly brilliant, and I'm left in absolute awe of the complexity of living systems. Every time I watch these videos, it leaves me with a warm, grateful feeling to be alive. This video is insane, such detailed animation that shows the process insanely well. This is also from 6 years ago, created for E.O. Wilson's Life on Earth, an interactive textbook of biology available free from iBook Store. Jesus, this is beautiful. This video is extremely cool, very, very good. Deserves more views and likes. Okay. So back at it here, the kinetochore is just like this structure that happens at the centromere, and is it happens in the metaphase. So the chromosomes continue to condense, and the nuclear envelope completely disappears. Then for metaphase, we have homologous chromosomes randomly assemble at the metaphase. So they randomly assemble. The microtubule is attached to the kinetochore, and where they have been maneuvered into place by the microtubules. Then we have anaphase 1. Spindle microtubules pull the homologous chromosomes apart. The sister chromatids are still attached at the centromere. Sister chromatids remain attached. Homologous chromosomes separate. Then we have the telophase 1 cytokinesis. Sister chromatids arrive at the poles of a cell and begin to decondense. A nuclear envelope forms. So we still have the formation of nuclear envelope during cytokinesis in telophase 1. A nuclear envelope forms around each nucleus, and the cytoplasm is divided by a cleavage furrow. Whatever cleavage furrow, this is what the cleavage furrow is. The result is 2 haploid cells. Each cell contains 1 duplicated copy of each homologous chromosome pair. Each cell contains 1 duplicated copy. So we had those homologous chromosome pair. We had those homologous chromosome pairs. Now each daughter cell from meiosis 1, like mitosis 1, is 2 haploid cells. Each cell contains 1 copy, not 2 copies, 1 copy. Then we have prophase 2. Sister chromatids condense. A new spindle begins to form. The nuclear envelope starts to fragment. The nuclear envelope in metaphase 2 disappears again, and the spindle fibers engage the individual kinetochores on the sister chromatids. In metaphase 2, sister chromatids line up at the metaphase plate. It's now sister chromatids. It's no longer homologous chromosome pairs. Then for anaphase, we have sister chromatids separating by shortening of the kinetochore microtubules. So the kinetochore microtubules shorten, and non-kinetochore microtubules lengthen the cell. Oh, so the non-kinetochore microtubules lengthen the cell. And for telophase 2 and cytokinesis, the chromosomes arrive at the poles of the cell and decondense. Nuclear envelopes surround the 4 nuclei. Cleavage furrows divide the 2 cells into 4 haploid cells. So you form 4 haploid cells from 2 haploid cells at the end of the meiosis 1 that all came from a 1 diploid cell. Diploid meaning it has 2 complete sets of chromosomes. At what checkpoint cells choose whether or not to divide? That is the G1 checkpoint, right? And P53 is in charge. Crossing over occurs in so it should be either it should be in metaphase, right? Oh, it's in prophase 1, crossing over. that's when they come together. This is where you have crossing over. It's in prophase 1. Metaphase is just randomly assembled. So that's where metaphase 1 is when you have the not crossing over. You have the independent assortments, right? States that two alleles. What states that two alleles of each gene become separated and only one allele from each parent will be passed to the offspring? Independent assortment, law of segregation. It says that two alleles of each gene become separated. So these two alleles of the parent become separated, and only one allele from each parent will be passed to the offspring. So an exception to this law is non-disjunction. Right. So what is an exception to this law? It's non-disjunction. Mitosis and cytokinesis occur in M phase. Female chromosome pairs are XX. We know that. Male chromosomes are XY. I mean, this is pretty easy. Let's do both as easy. What refers to genetic conditions associated with mutations in genes on the X chromosome? So genetic conditions associated with mutations in genes on the X chromosome is X-linked. Recessive inheritance refers to genetic conditions on the X chromosome, X-linked, recessive. Carrier mother. So we have unaffected father, we have carrier mother. So the purple is what causes that. So you do XX, we have a daughter, so it's unaffected. We have XY, unaffected, and this X with the Y is what causes the son to have the disease. So it's X-linked and it's recessive. And we have the carrier daughter, and that's when you have the bad gene, the one with the disease, with the X from the father, which causes the carrier daughter. And in males with an X-linked recessive mutation, the mutation will or will not be expressed. So in males with X-linked, the mutation will be expressed. This is because males only have one X chromosome. In females with X-linked recessive, the mutation will not. Females have two chromosomes, so the mutation is carried but not expressed. The normal X chromosome will be expressed because this is a recessive trait. What are famous examples of X-linked recessive disorders? Hemophilia, factor 8 deficiency, is a bleeding disorder where there is a deficiency or dysfunction of clotting factor 8. Prolonged bleeding, easy bruising, and joint bleeding. Hemophilia again. Duchenne muscular dystrophy, DMD, progressive muscle wasting disorder caused by mutations. and the dystrophin gene. Muscle weakness, difficulty walking, respiratory problems, color blindness is a condition where individuals have difficulty distinguishing certain colors due to a lack of dysfunction of photopigments in the eye. Lack of photopigments, difficulty seeing. This is like it should be with cons, right? Not rods. Difficulty seeing specific colors. Genes for red and green color receptors on the X. And muscular dystrophy, Becker muscular dystrophy, BMD, is a milder form of muscular dystrophy. So yeah, it's important to note that carriers of X-linked recessive disorders, heterozygous females, typically do not manifest symptoms due to the presence of a normal allele however, they have a 50% chance passing a mutated allele to their sons, who may then express the disorder. Interphase includes what cell cycle phase? It's all G1, G2, and S. So G1, S, and G2. What gene is responsible for initiation of male sex determination and codes for testicles? XY, the Y gene, the SRY gene, sorry you're a male. So it's responsible for initiation of male sex determination and codes for testicles. We have the short arm, centromere, and then the long arm, the sex determining region, SRY, sex determining region Y. This gene is Y-linked because it is found only on the Y chromosome. Semen is a combination of sperm and seminal fluid, right? Sperm from production to ejaculation. Sperm develop as tiny round cells in seminiferous tubules. Hormones from testicles and pituitary gland help sperm mature. They travel to epididymis and develop further for several weeks, migrate to vas deferens, travel to seminal gland where nourished by semen fluid, travel to prostate gland and mix with prostate fluids, exit through urethra during ejaculation. Amazing. What makes viscous fluid to clean out the urethra? We know this is the bulbourethral gland that does this. Yes, makes viscous fluid. The bulbourethral gland is also called the corpses. What was the name? It's probably a scientist. Cowper's gland. Cowper's. How do you say that?
Cowper's.
Like cow, right? You get it. What make alkaline fluid to help sperm survive the acidic environment of the female reproductive tract? I believe so we have the 7-up, the seminiferous tubules, the epididymis, we have the bulbourethral gland. I think it's bulbourethral gland and something else, the seminiferous tubules, seminal vesicles, and prostate gland. My bad. So we have it here, right? We have the prostate gland and seminal vesicles. So this is before the 7-up, right? The pathway. So seminal vesicles. Vesicle is like a gland, right? It's like a cyst to make alkaline fluid to help sperm. So where are we here? Like this is the sperm prostate under the bladder, and we have the seminal vesicles here. Then we have the bulbourethral gland, which is after that, where you need to. What does bulbourethral gland do again? It makes viscous fluid. to clean out urethra of urine, right? So we have the testes here, we have the urethra, we have the bladder, we have the vas differentia, we have the vasectomy, where you are cutting the vas deferens, and we have urethra. Okay, why do people do vasectomy? Is it like testis cancer or something? Why do men do vasectomy? It's to achieve permanent male sterilization, making it an effective method of contraception by blocking the vas deferens. Sperm are prevented from reaching the semen ejaculate during sexual activity, resulting in infertility. But like the deliberate use of artificial methods or other techniques to prevent pregnancy as a consequence of sexual intercourse, the major form of artificial contraception are barrier methods, of which the most common is the condom, contraception, which contains synthetic sex hormones that prevent ovulation in the female, intrauterine devices such as the coil, which prevent the fertilized ovum from planting a male or female. Okay. Family planning, convenience, the relatively simple, quick procedure compared to other forms of permanent contraception, such as female sterilization. Recovery time is usually shorter. Effectiveness, pregnancy-related complication. Reverse insomnia, the reversal is not always successful. But why? Like, is it due to disease? Why do they want contraception? People use contraceptive birth. Spacing births, delaying parenthood, maternal health, strengthening. Okay, is it all like psychological? Like it's for relationship? I thought it's for disease. Is it for a disease too? IUDs can be used to manage conditions such as heavy menstrual bleeding and Okay, apparently not. There was another question I had, which was does the male ejaculate after vasectomy? Yes, it does not affect the process for the man continues to ejaculate during sexual activity, but the ejaculate semen will no longer contain sperm. The purpose of vasectomy is to prevent sperm from being present in the semen. So the sperm travels through the vas deferens, right? To come from the epididymis here to the ejaculatory duct. Okay. Nice. So this we didn't get it. We do it again. Pathway of sperm during ejaculation is seminiferous tubules, epididymis, vas deferens, ejaculatory duct, urethra, and penis. What have follicles that produce ova? It is the fallopian tube. Ovaries produce ova. My bad. They have follicles that produce ova. What is the production of female gametes? So what are follicles in ovaries? Are small fluid-filled structure found in the ovaries of females. They are small and fluid-filled. They consist of immature egg, oocyte surrounded by specialized cells. Okay. Here's an overview of the role and types of follicles in the ovaries. Okay. What is the production of female gametes? Oogenesis. Spermatogenesis and oogenesis. How are sperm and eggs produced? We have oogenesis, oogonium, spermatogonium, puberty, old age, from puberty to old age. Then we have primary oocyte, we have primary spermatocyte, secondary spermatocyte, secondary oocyte, we have egg cell ovum, we have stage 2 of mitosis, we have spermatids, spermatozoa, motile sperm or spermatozoa. So from puberty to old age, we have spermatogonium. Okay. What develops the female reproductive tract and thickens the uterine wall? It should be, so developing is estrogen that establishes the female, but progesterone is for promoting growing it. So estrogen establishes, progesterone protects the endometrium. Estrogen responds to FSH and LH does for progesterone. Estrogen affects, builds up uterine lining, increases body fat, depression, headache, migraine, interferes with thyroid hormone, increases blood clotting, decreases libido, impairs blood sugar control, increases risk of endometrial cancer, increases risk of breast cancer. But progesterone maintains uterine lining, secretory, helps use fat for energy, it's antidepressant, facilitates thyroid hormone action, normalizes blood clotting, restores libido, regulates blood sugar levels, protects from endometrial cancer, probable prevention of breast cancer. So estrogen is basically bad, it's bullshit, we don't want it, but progesterone is really good, feels good. Female egg pathway. So we do have ovary to the fallopian tube, to the uterus, to the cervix, to the vagina. So from ovary to the fallopian tube, to the uterus, to the cervix, to the vagina. If unfertilized, the egg will leave through the cervix and vagina, otherwise it implants into the uterus as an embryo. So when it's unfertilized, it is menses that happens, and during menstruation, the shedding of the endometrial wall happens. Cool. Fertilization occurs in what? Fertilization occurs in the endometrial wall, the fallopian tube. Let's do that again. So we have, this is a good one. We have the ovary, you have the mature follicle, developing follicles, we have ovulation, we have fallopian tube, fertilization. If it happens, if it doesn't happen, so when it happens, we have the zygotes, we have the cleavage, we have the blastocyst, morula, blastocyst, implantation beginning, uterine wall, uterus, amazing, cervix, vagina. Okay, nice. It gives a really nice picture. What organs are controlled by FSH and LH? So, yeah, I think it's ovaries. Yeah. So we have oogenesis and spermatogenesis, fish eggs. We have the fish, FSH. We have anterior pituitary, antique picture. What the hell? Antique picture. These fish oocytes, FSH. Then we have testosterone, estrogen, photo of a girl. We have the testosterone. This is like Captain America, right? Test tube. We have LH, lieutenant. So lieutenant, luteinizing hormone, LH, testosterone, estrogen. So, but estrogen is a luteinizing hormone, but LH is a luteinizing hormone itself. So we have FSH with progesterone, estrogen response is a response to FSH, right? The process of fertilization involves the following 4 steps. So probably we have, the eggs that come out of the ovaries, and then we have the oocytes that go to the fallopian tube, and then if they fertilize, they become zygote. The zygote differentiates. It gives multiple cells that goes to uterus and implants in the wall. So let's see: sperm binding, acrosome reaction, cortical reaction, genetic transfer. Overview of the process of fertilization. So we have the sperm head here, basal body, centriole. We have acrosome, so sperm binding, and then acrosome reaction. So we have the sperm binding jelly coat, sperm binding receptors. Boom. The hydrolytic enzymes are here. Hydrolysis means they break down the water, right? Like the gel. And then we have vitelline layer, whatever that is. Egg plasma membrane. So acrosome reaction, and then what reaction? Sperm nucleus goes into that acrosomal process. Sperm plasma membrane, fused plasma membranes. We have cortical reaction and genetic transfer. So cortical granule, perivitelline space, fertilization envelope. Okay. Crossing over occurs in, so the recombination happens in prophase 1. Yes, synapsis, prophase 1. What states that 2 alleles of each gene become separated and only 1 allele from each parent will be passed to the offspring? So what is an exception? So this is law of segregation. An exception is non-disjunction. Let's remove the law here. So law of segregation is basically only 1 allele from each parent passes on to the offspring. What gene is responsible for initiation of male sex determination? So male sex determination is SRY gene, sex determination, sex determining region Y. what make alkaline fluid? It's the prostate gland and the seminal vesicles to help the sperm survive the acidic environment of the vagina, a female reproductive tract. What have follicles that produce ova? It's ovaries. Female egg pathway is the female egg pathway is from ovaries to fallopian tubes to uterus and to vagina. Cervix and vagina. So uterus, cervix, and vagina. Okay, nice. We finished chapter. Let's rename it. Bio B2. Is it reproduction, or is it yeah, reproduction. Chapter 3 is embryogenesis and development. Sure. Reproduction. Okay, I'll see you in the next video. Peace.